Today, I will talk about understanding the pathogenesis of JAK2V617F positive MPNs and single-cell resolution. Myeloproliferative neoplasms are a class of diseases which are characterized by overexpression of one or more mature myeloid blood cell lineage. MPN often occurred in the aged population. It has a risk to transform to the acute myeloid leukemia. Before the transformation, it stays in the MPN period for a long time. Thus, MPNs can act as one of the ideal disease model to work for the pre-leukemia, to study the pre-leukemia disease. In 2005, several groups have identified that in the Philadelphia negative MPNs, they have the common acquired somatic JAK2V617F mutations. You can see here that they are greater than 90% of the PV patients carry JAK2V617F mutations, and also greater than 50% of the ET and the PMF patients carried these mutations. Then, in detail, in genetic level, it's G to T substitution result in the amino acid switch from V to F, thus leading to the constitutive activation of this JAK-STAT pathway. We know that JAK2 can couple to the several of key hematopoietic signaling receptors like EPO and TPO. That is why finally it can result in the excessive production of red cell platelet or neutrophils, leading the different MPN subtypes. The fundamental question in the field still will be why a single mutations will result in 3 different MPN subtypes. Previous studies have shed important lights on the heterogeneity of MPN phenotypes. For example, in 2015, Dr. Green's lab has found that the presence and order of additional somatic mutations can affect the final outcome of the disease phenotype. For example, if the patient have JAK2 mutation first, followed by JAK2 mutation, they have the high chance to develop of ET. Otherwise, it can develop in PV. Also, the germline genetic variation also can predispose the disease phenotype. The clinical observation and my study support that if the JAK2V617 allele burden is higher and its homozygosity, which is often associated with a PV phenotype, while the lower allele burden and heterozygosity often associated with a ET phenotype. Despite these tremendous advances, it's still, for most of the patients, we didn't understand why the one single mutation causes 3 different phenotypes. But we do know from the transgene mouse model, from the transplantation, from the MPN patient study that the mutations is occurred in the very hierarchy of the hematopoiesis in the HSC levels. Then initially, we just curious about the transcriptome consequences if we have this mutation, if the patient have these mutations. To do that, we isolated JAK2V617F positive PV and ET patient bone marrow cell, and we isolated a pure population of HSC and erythroid progenitor cells. And we got around hundreds of cells. We use it for the low input RNA-seq analysis. Interestingly, we found that both in HSC and MEP levels, the controls, PV and ET patients, they show distinct transcriptional landscape. And within the ET featured gene clusters, we found that one of the signature gene is VWF. What is VWF? In 2013, first time it has shown that in the HSC, they have a subpopulation is VWF positive HSCs, which are Mk primed HSC. They can directly differentiation to the Mk progenitors, the mature into the megakaryopoiesis. Then the high expression of VWF in ET HSC might suggest that the ET HSC have a strong Mk priming potential. To test this hypothesis, we use the two Mk primed gene sets. One is Mk progenitors. The other is from the state-state hematopoiesis. There is a single-cell RNA-seq analyze that exists the Mk primed HSCs. Used both gene sets, we consistently found that the Mk primed gene is highly expressed in ET HSC, but not PV HSCs. In 2015, another study showed that in the inflammatory stress, CD41 positive HSCs, they are stem cell like MKP, which can differentiate to the progenitor MK and megakaryopoiesis, and it could be differentiation from VWF positive HSCs. Then use these two populations as the marker, these two genes as the marker. We look at that the Mk prime, the HSC population changes in the patients. We found that the VW positive HSC and the CD41 positive HSC are uniquely high. The population is expanded uniquely in ET HSC, but not PV HSCs. To understand the molecular level of the underlying biological processes or signaling pathway, we performed the GSE analysis. We found that it's the interferon alpha response is very uniquely enriched in ET HSC, but not in PV HSC. And the molecular level is key component state 1 as well as the phosphorylated state 1, but not other states are highly expressed in ET HSC. More importantly is if you look at that, the contribution of why the phosphorylated state 1 is high, we found it's mainly contributed by the CD41 positive Mk primed HSCs. When the HSC differentiation to the MKP is higher, they still maintain the higher inflammation signaling compared to the PV MEPs. So we found two features from the bulk HSC analysis that ET HSC first they show that high MK priming potential. Second, they show the higher IFN signaling. However, we know that the pathogenesis, the JAK2V617F mutation is a phenotypic driver for the ET pathogenesis. How could we link the mutation with the HSC heterogeneity and the IFN signaling? They already have two well-developed techniques that can couple the single-cell RNA-seq with the mutation detection in the same cells. And these two technologies have already shed important lights in the JAK2 positive myelofibrosis and color mutant MPNs. So we just adopted the target-seq method so we can identify the ET HSC into precisely identify them as homomutant, heteromutant, as well as non-mutant cells. When we compared the ET HSC with different genetic mutation background with normal HSCs, we found that they indeed expressed very high platelet and MK-related signature genes. You can see plaque VWF CD9 is highly expressed in the homomutant cells. Besides highly expressed these genes in each individual mutant cells, we also found that the Mk primed population is enlarged. You can see if we look at the VW positive HSCs, also if you look at the common genes to define the Mk primed HSCs, they all consistently support that this population is enlarged. Why the population is enlarged, we found that it's because the mutant confer the cell high proliferation capacity and their cycle is very active. The behind molecular mechanism could be that the strength anti-apoptosis mechanism as well as the activate PI3K mTOR signaling pathway. And to understand that why the mutant cell Mk primary potential is enhanced, we look at that the altered signaling pathways. We found that these in the mutant cells, in general, they highly express the inflammatory related associated signaling pathway, especially the interference. So we can see that they're highly in the mutant cells. Also, the key component is state 1. Thus, our data links IFN signaling with JAK2V617F mutant ET HSC and diagnosis. Paradoxically, that IFNs also is the mainstream therapeutic approach for MPNs. It has used for treatment MPN for over 30 years. And it shows that clinical, hematological, histopathological, even sometimes molecular response in the clinical studies. Then the dual effect of IFNs during the disease pathogenesis or the disease treatment, how could we understand this phenomenon? To do that, we just tried a wide dosage of IFN alpha and gamma as well as a different treatment period and used the CD34 hematopoietic stem and progenitor cells. When we induce the differentiation to the megakaryopoiesis lineage, we found that low dosage and short-term treatment indeed stimulate the CD41 positive cell generation, while high dosage and long treatment repressed this population, these cells. And when you use the combined treatment for short time, and we can see even stronger induction of these CD41 positive cells. But we still cannot distinguish its the stem cell effect or its progenitor effect. So we purified the HSCs, performed the same stimulation. We found that and we conduct CFU colony assay. We found IFN gamma and alpha and gamma combined treatment indeed directly stimulate HSC to generate Mk colonies. Next question will be, we know that the HSCs in ET are heterogeneous. They include the mutant cell, also the non-mutant cells. Then how these two different groups of cells respond to IFN? To do that, we isolated the patient HSCs into single cell. Then we let this single cell to grow into clones. Then half of the cells from these clones used for genotyping. Then we can see whether these cells is mutant or non-mutant. The other half we used it for with or without IFN treatment. During the first phase, we found that cells HSC from patient that displayed the strong proliferation capacity with especially the mutant cells contribute very high clones. And during the differentiation, we found that IFN treatment indeed stimulated more Mk colony formation in the mutant cells we compared to the non-mutant Y-type controls. This result suggests that the mutant cells they exhibited the higher responsive or higher sensitivity to the low dosage short-term IFN treatment. Also, interestingly, when you see PV, you can see that they display an opposite trend, and they kind of repressed by this short-term IFN treatment. So it means that this is a unique property for the mutant ET HSCs. To understand why these cells to respond the most sensitive to the IFN treatment, we look at that they alter the signal pathway. We found biological processes and pathways. We found that these cells, they exhibited very active metabolic processes. Then we linked that it's because that the mutation or the IFN stimulation changed their metabolic process. Then they can provide the basic metabolites as well as the energy required for the proliferation and activation. That is why these cells can be more active upon IFN treatment. Then single cell RNA-seq gives two features of the ET HSCs. The mutant ET HSC, first is they showed the high Mk priming potential. Second is they have the high IFN response. So we propose that it's possible that ET pathogenesis might happen its JAK2 mutation in this Mk primed HSC with enhanced IFN signal in this population is enlarged and the potential is become strong. Then they can more efficiently to differentiation to the megakaryopoiesis lineage generated excessive platelets. But we didn't rule out another possibility is somehow IFN signaling is enhanced in ET patients, leading to the enlarged disease population. Then again, the beta JAK2V617 mutation in this Mk primed population may further facilitate the platelet generation. As I mentioned that IFN still is the mainstream therapeutic approach. Then how about after chronic therapeutic dosage IFN treatment? What about this mutant HSC? What are their behaviors? Previous study in the MPN disease mice have shown that IFN can directly target these mutant stem cells. They either induce the apoptosis in these cells, or they just stimulated their cell cycle finally to induce the exhaustion. But all these studies, it's currently still limited in the mice. We still don't know that in the MPN patients, what happened for these HSCs after treatment. To answer these questions, we collect patients either treatment with single IFN or combined with hydroxyurea. We isolated their HSCs and performed the target-seq. First, we found that the Mk primed potential is significant. The population is reduced, preferably influenced of the HOMO mutant cells. Second, we found in each individual cells, the HOMO mutant cell Mk potential is significantly reduced. But interestingly, we found in heteromutant it increased. And to see this different behavior of the different mutation cells, we look at what happened in the molecular level. We found that first, after treatment, the commonly, no matter is HOMO or heteromutant, the IFN response, they all increased. But when you look at the cell cycle, the HOMO mutant is reduced, the hetero is slightly increased. Apoptosis level, the HOMO is reduced and hetero is HOMO is reduced, HOMO is reduced and hetero is induced. It suggests that the hetero cell is go to the apoptosis, but what about the HOMO mutant cells? We go through the GSE analysis used the entire hallmark GO terms. We found that over 95% of the GO terms in the HOMO mutant cell are downregulated, especially that displayed very low metabolic activities. Then these results lead us to hypothesize that the HOMO mutant cells are forced into quiescence after treatment. Then finally, we give up proposed models that it's after treatment, the HOMO mutant cells, they're forced into quiescence, while the heteromutant cells, they are induced to the apoptosis. This might can explain that why clinically, when you discontinue of IFN in some patients, you can see very rapid molecular relapse. Maybe the cellular basis is this sleeped HOMO mutant cells without IFN, they just wake up and they relapse, they contribute to the disease relapse again. And This work is a collaborative project in my lab, Dr. Zhang, Dr. Chen, and Dr. Wang's lab. It's mainly finished by Jingyuan, Yanhong, Sunqing, Makai, and Shi Hui. I also need to thank for my other collaborators, Dr. Zhu, Dr. Zhou, and Dr. Gruen, as well as lab member in his lab, Jia and Stephen, as well as my collaborator, Bresnik. Thanks for my funding support. Thank you for your attention, and